Hello, and welcome to this video on artificial yeast chromosomes. These are genetically engineered chromosomes produced in a lab. These can be disassembled in bacterial plasmids that can be cloned efficiently and reliably, insomuch that they produce a procedure that both works and does so in an expected manner for a low cost. Due to stability issues, these are now considered obsolete. This video will build on the genetically modified yeast video linked in the description box below. These yeast cell lines rely on a replication feature of yeast cells. This is known as the autonomous replication sequence. This was a precursor to the plasmid vector. This produces a stable gene or DNA sequence which has telomeres on the ends and a centromere. These are structural aspects missing from intracellular nucleic material. As a consequence, nucleic material within the cell that lacks these features tends to be broken down and dismantled by enzymes very quickly. Yeast artificial chromosomes are essential to the development and use of genetically modified yeast strains and the development of better strains of brewer's yeast. The yeast artificial chromosome is slightly different to normal yeast DNA and plasmids, in that it is linear. It can be incorporated as part of a vector, but this requires modification. Firstly, the removal of the telomeres, and later the centromere, and then finally addition of several gene factors such as selective genes and the excising and incorporation genes needed for plasma insertion into the genome. The benefits of yeast artificial chromosomes is the ability to clone 50 to 2000 kilobase pair stretches of DNA. These are large gene sequences and sequences of genetic material. They are far too long for conventional bacterial cloning methods. Unlike DNA that is cloned independent of the cell, these chromosomes are stable throughout production. They can also be reintroduced into mammalian hosts and incorporate into the host DNA, or not into the DNA, and still be used and expressed by the host. There they function as per normal, as noted by Geraldo and Company 1999. Variegation and inherited somatic mosaicism have been observed in transgenic mice carrying yeast artificial chromosomes, in which a DNase, one hypersensitivity site, located 1 to 12 kilobase pairs upstream of the mouse tyrosinase gene, had been deleted. At present, we are generating new transgenic animals with minor deletions of the HS or hypersensitive site. This is a good example of how a gene can be taken from yeast and put into an animal and still be expressed. A method for linking any standard yeast artificial chromosome is described by Laron and Company in 1996. The yeast artificial chromosomes are introduced into the same cell and joined by a mitotic recombination between the different arms of it. This creates not only a single sequence, but a homologous single sequence that links each of the components of it. Several of these chromosomes can be recombined, and this can be done in order, meaning a particular gene can be used to activate a secondary gene. This uses a chain of vectors that were selected by the researchers or developers. The linking can also contain the relevant genes that are needed as an expression reporter in mammalian cells, as an example, beta-galactosidase. Unfortunately, the process of cloning yeast artificial chromosomes is plagued by random mutations, lost base pairs in the form of deletions and rearrangements, and other negative. As a result, creating it is relatively simple. Maintaining it is relatively simple. Cloning it is not. And this means that it can only be used in a small population or transferred to a small population. The advantages of yeast artificial chromosomes are the previously mentioned transition between mammalian hosts, the stable production, and the ability to build a sequential genome out of it. This can also be used in reverse, to dismantle a genome and understand what it is made from, and this is referred to as walking. As can be seen here, it is used in the construction of mouse genome assays. To produce better industrial yeast strains, whether for brewing medicine or research, is a little bit different. For example, S. cerevisiae, this is also better known as your standard brewer's yeast. It is also being used in the bioethanol industry and for production of heterologous compounds such as human insulin, the hepatitis vaccine, and the human papillomavirus vaccines. Steensel and Company present a simple diagram of the ways yeast is improved across industry and research. The most relevant to the home brewer is mutagenesis and genetically modified organisms. For the researcher, it is genetically modified organisms and hybridization. This quote from Steensel is the best summation of why GMO yeast is the best method for improving yeast. Strain improvement by genetic modification differs from the non-GMO approaches described above in two important characteristics. Firstly, the DNA of the organism is changed in a specific, predetermined manner without changing other parts of the genome. This implies that in theory, one or several phenotypes can be changed without negatively affecting other important characteristics. This reduces the chance of creating crippled strains. Second, the technique is not limited to naturally occurring or randomly induced mutations. Brewing and other industries in the 80s and 90s adopted the principles of recombinant DNA technology, and they developed new, improved strains for industrial purposes. This often used the GM techniques that have been described elsewhere. These experiments were focused on single genes and enzymes, and their aim was to manipulate these in isolation to create the product of interest, for example, an improved ethanol tolerance. The main targets for modulation of those enzymes, transporters, and regulatory proteins had this effect in some cases. The opportunities of metabolic and genetic engineering included adjusting or fine-tuning the number and type of genes activated, the level of gene activity, the gene expression, and the protein or enzyme activity, and in some cases, even the protein at a subcellular location and level. Apart from the regulation at the level of transcription, proteins also affect the translational efficiency of the genes themselves. This in turn can be influenced by the use of codons. Codons produce protein. Codon optimization, particularly of heterologous genes, can then have an effect on the gene itself, but it can also have an effect on how efficiently proteins are created. And this is particularly important in the glycolytic genes and pathway of cerevisiae. By significantly increasing the protein levels, this could theoretically significantly increase both the tolerance of yeast to ethanol and to other factors. Another novel and surprisingly powerful tool in the development of industrial yeast strains is that of creating a synthetic biological yeast. Broadly speaking, this has the design of creating an entirely non-naturally occurring molecule or structure within the yeast, or having the yeast produce a completely new and not naturally occurring compound. Arguably, examples of this include the opioid yeast mentioned in the earlier video. By taking single genes from one organism to create a completely new organism, or to redesign existing natural biological systems, these can be considered synthetic biology, and they can assemble more complex genetic systems, and in the end, have a significant effect on the metabolic pathway. More than that though, there's another project called Synthetic Yeast 2.0. This is seeking to reconstruct and redesign the full genome of cerevisiae. This is going particularly quickly and yielded a very recent publication of the first partially synthetic eukaryotic chromosome. Environmental conditions during fermentation or propagation are also harsh, and this is another way in which genetically modified chromosomes and yeast in general can be very useful. They are often faced with drastic changes in the substrate, the environmental conditions, and the way in which they are stored. They must respond quickly to this, or the cell will die. As a result, industry's major focus, along with everything else, is to improve these characteristics and how well the cell can express or activate the processes that are needed for them. As an example, Perez and company in 2010 identified pathways responsible for osmotic tolerances, and the overexpression of these genes exerted a benefit on the yeast performance, in this case leavening bread. In a different study, they improved the freeze tolerance of baker's yeast, and they did this by disrupting certain genes, or by replacing the wild type of the gene. By modifying these, they had less sensitivity to feedback within their environment, and this meant that they could freeze better. This is important when you consider things like stuck fermentations. These are a common issue, particularly for the home brewer. When these fermentations become stuck, it's often a lost cause, and the entire brew must be thrown away. In an industrial setting, that's really not a very viable option. As a result, a stuck fermentation when producing bioethanol would be a significant loss of money, time, and effort. Mechanisms to account for and improve the probability of the wash completing on time and in a manner desired by both the home brewer and industry are therefore very desirable aspects of this form of research. That, along with increasing yield and/or productivity of a yeast strain, the ability to recycle yeast, or the ability to use it across multiple industries and multiple roles. Although obsolete, yeast artificial chromosomes were, and in many ways continue to be, an important first step in the development of genetically modified yeast. This yeast has many roles to play in the future, and it plays many roles now that are important to daily life. Thank you for watching this video. Please post any comments, questions, or suggestions you may have below.